This is going to be the third page of vocabulary words. Let's start off with gel electrophoresis. Another way to say this is DNA fingerprinting. Now this is going to be sorting DNA by size and charge. Usually size is what you're going to see, and you're going to look at the gel. They will be the wells at the top, and then you're going to see the bands down towards the bottom. Smallest bands travel the farthest and the fastest. They're at the bottom. Largest bands are at the top. Now what we're going to do is you are going to look for an evolutionary relationship. You're going to look for a familial relationship. You may look to see if a species is related. Solve a crime scene, put someone at the crime. Those are all different reasons why you would look at a gel. And you would always say the most bands in common if you're going to be writing a short response and you need your proof to be able to show support for your answer. Species X and species Y are more closely related because they share the most bands in common. Botanus curus and species Z are related because they share the most bands in common. Okay, next word is gene manipulation. Another way to say gene manipulation is going to be genetic engineering. So what you're actually doing right here, you are taking a gene and you're going to insert the gene into a new organism. You don't have to know this word, but the new organism, the organism that has that name is now going to be called transgenic. Right? Trans, we've changed the genes. Transgenic. Trans meaning to change, and genic meaning the genes. We've changed the gene. So if I inserted a gene into salmon to be able to swim longer or to be bigger, whatever it is that I want for the salmon to have, that will be then considered a transgenic salmon. Now, we would do this because we would want this to happen quickly. The reason behind this is because this is much quicker to have done. Anytime I'm going to do gene manipulation or genetic engineering, I would insert the gene into bacteria. So if I were to look at it as bacteria, here's your bacteria. And the bacteria have little rings of DNA. That little ring gets taken out. That little ring gets cut right there. And here's the new gene. Then what you wind up having, that plus that, and then you have this new DNA with inserted. So that's new DNA with inserted gene. And then that goes back into the bacteria. Then that bacteria, whatever organism, makes that identical protein that's coded for on that gene, whatever it is. If it is human insulin, that bacteria is going to make human insulin quicker. If it's human growth hormone, that bacteria is going to make human growth hormone quicker. All right, so we have transgenic organisms, and then we have a bacteria that can do the same thing. All right, next one is going to be glucose. Glucose, we can say, is a high energy molecule. It gives us quick energy. It is the product of photosynthesis. Now, the most important thing that we need to know about glucose is that energy is stored in the bonds of glucose. You are going to see that question as far as the bonds of a molecule. Energy can be stored. You can get energy from protein. You can get energy from digesting anything. If you need energy, your body's going to get it. It just is that glucose is your quick energy. And fats, lipids, they're your slow, long-term energy. And if I needed to, I can get energy from protein as well. But we're going to use proteins for other things. It's usually last resort when there is no fat or if there is no carbohydrate to use for energy. All right, next one is a guard cell. The guard cells, remember, they look like little lips. They're like that. That's an open guard cell, closed guard cell. That right there, that opening, is the stomata or stomate or stoma. Any one of those words works. And the whole point about the guard cell is regulation. That's the life process that it's taking care of because it regulates water loss. That's how they open and close. This happens in plants. So if it's raining a lot outside, that stomata will open, the guard cells will plump up, and then water can leave the plant. When it's dry out, the stomata, the guard cells get thinner, and then the vacuole inside it loses water. They get thinner and they close up, and then what happens is water stays inside. So the guard cell is specifically on either side of the stoma. All right, HIV is the next one. This is human immunodeficiency virus. I'm writing very sloppy. HIV, human immunodeficiency virus. Basically compromises the immune system. Okay, I'm going to restart in 3, 2. Next one we have is HIV, which is human. There's the H. Immuno. I'll make it capital I. Immunodeficiency. And then virus. That's the V. Okay, so human immunodeficiency virus. Basically, it compromises the immune system. The basic thing that you need to know about HIV is that it stops your own immune system from working. So your immune system cannot fight off a pathogen. So people don't die from HIV. Because your immune system can't identify friend or foe, they can't identify the pathogens, your white blood cells can't function, nothing works. So what happens is you could die from like pneumonia. So that could be the problem there. So it's the human immunodeficiency virus. The virus basically attacks your immune system. For the most part, it shares the same cell receptor that your immune system would use. So it stops your immune system from actually functioning, which is kind of crazy. You really can't maintain homeostasis in that sense because you can get sick by something else. And homeostasis is our next word. So maintain, homeostasis is maintain internal balance. All body systems work together to maintain homeostasis. To maintain homeostasis. Our body systems, they work together to maintain dynamic equilibrium. They work together as feedback mechanisms to maintain homeostasis. That's our biggie. Okay, failure to maintain, which we all know, failure to maintain results in illness or death. Illness first. I don't know whether we're an S there, illness, and then death. All right. Next one should be insulin. Okay, insulin is a hormone, which means that it is a protein, which means that it is a chemical, means that it is a molecule. Those are all the words that you could see that describe insulin. The main function of insulin is to lower blood sugar. And insulin is part of a feedback mechanism. So if you have too much sugar, your body releases insulin. It tells your cells to absorb that sugar from the blood. And it maintains a certain range. So what you'll see here is you'll see this will be your blood sugar, and it does this, and then we want to maintain homeostasis, maintaining balance right there in the middle. Usually you'll see time on the bottom, and you'll see blood sugar on the side, and it'll say, oh, when was insulin released into the bloodstream? And you'll have to know that this is when insulin is released in the bloodstream because that's when the blood sugar goes down. Now when it's low, you have another hormone that's released to release sugar back into the bloodstream, and the cycle goes up and down. You are also told that you're hungry, so that way that you put more sugar in. All right, next thing is a limiting factor. To limit something is to stop. And a factor is something in the environment that can make this happen. So we're going to say something that caps the size of a population. What could cap the size of a population? Right off the bat, food. You don't have enough food, your population can't continue to grow. People start to die. Same thing with water. You don't have enough water, people start to die. Now, we don't have any predators, but predators in the wild, if I have a lot of predators in the area, I am not going to have a lot of, whatever that predator eats. That predator is going to cap or limit the size of that population. It could also be sunlight. Sunlight is something that could limit the size of a population if you're a plant, right? So now if we look at these things, 2 of them are biotic and 2 of them are abiotic. You should know that if I draw a line like this, everything here at the top is biotic and everything down here is abiotic. That's always going to be important to know. When you talk about these words, then you have to know the vocabulary word and then know an additional vocabulary word as well to be able to answer those questions. Next one is the lysosome. Lysosome, now you need to know that it is an organelle. It also may be written in the Regents exam question as a structure. Which cell structure performs this role? Well, lysosome itself, it's responsible for digestion in a cell. Because it's an organelle, it's not an organ, it's teeny tiny, it's within a cell. And it is responsible for digestion. It helps to break things down. So it can break things down, make them tiny. If the cell needs to reuse something. If the cell needs to dispose of something that needs to get through the cell membrane, it can help with that as well. Okay, next word is meiosis. Remember, meiosis makes your sex cells. What happens in meiosis, we will go from 2N down to N. We are now haploid. It reduces chromosome number. And this is for sexual reproduction. You're making gametes, and they are sperm and egg. When you see it, we start off at 2N, and we get broken down. And the end result in each one of these is going to be N. So if I start off with 8, then I end up with 4. Whatever it is, humans, we start off with 46, we end up with 23. Now, for the sperm, I get 4 functioning sperm cells. For the egg is a little different. I'll do this in a different color. The egg, if I start off with 2N and I go down, and I go down again, I get 1 big egg, still N. These leftovers are just to get rid of. It's a place to put extra chromosomes. Remember, we talked about an egg. The egg has to have all the organelles. It needs enough energy stored to be able to start reproducing. Okay, that's very important. Next one I have should be mitosis. Now, mitosis, we're going to make body cells. Another name for a body cell is a somatic cell. This is different. I am 2N and I end up with 2N. Identical DNA. So identical genetic information. It's not necessarily going to be identical cell, because what if 2 mitochondria are on one side and 1 mitochondria is on the other? Be very, very careful with that. It's identical genetic information. The DNA is going to be the same. We are 2N going to 2N. That's what's the same. This is basically for growth and repair in multicellular organisms. And then for reproduction in single-celled organisms. Or if we're talking about asexual reproduction, the cells will split in 2. So depending upon the type of asexual reproduction, you can have different size cells. But make no mistake about it, the DNA has to get, the DNA comes out copied identical. All right, that's it.